So it's a pleasure to have this special seminar today with Professor Hiroki Shirai. He's a professor now he's a professor at university, visiting professor at university, and he's a scientist at Riken, which is sister, brother, partner to BSc. There's always been a lot of good collaboration between BSc and Riken, and I know that both directors from both centers are always trying to fostering that. So we will really try to initiate some more work together. It's great because he's one of the world experts in antibody design, and especially from a structural point of view. He has been participating in these competitions of generating a structure of antibodies, and with excellent results. And something I particularly like a lot is that he also adds a lot of industry point of view. He's been in Stella, pharmaceutical company from Japan, for 17 years almost. Yeah. Almost 17 years. So and then he just moved in the last couple of years to Riken and to reconnect with academia a little bit and bring things down into. So it's a great chance for us. Also, he has been 1 year in Cambridge with Tom Blundell, which is one of the excellent labs. So I think he's going to be talking a lot about how artificial intelligence is making impact on this antibody design. So we're very excited to listen to your talk.
Thank you very much. Thank you, Professor Gerard. So I'm very. I would like to express my gratitude to Victor-san and everyone involved in the seminar and everyone in BSc, right? So I'm Hiroki Shirai, a coordinator at the Riken Center for Computational Science, abbreviated as RCCS, which is a similar organization with Barcelona Supercomputing Center. Okay. This is the first time to visit BSc, and as well as the first time in Spain, all right? So I'm very happy to be here. But anyway, so that today the title is AI and Simulation for Antibody Drug Discovery, okay? My talk today has two purposes, all right? First, I will discuss how the business model for using in-situ approaches in antibody drug discovery has been evolving, sharing recent examples from our own research, right? So yes, I took a read of this field when around 10 years ago, okay, based on the UK. And at that time, using the framework of industry academic relations collaboration, we construct the broad view of business model in terms of antibody information drug discovery. So we summarize the flow of fraud and obstacles, solutions, and the science, because interests differ depends on the positions. Okay, so I would like to tell you in detail later, but anyway, this is the 10 years ago's model. Now, dramatically changes in terms of the needs and the solutions, right? Needs has changed to antibody engineering is much more important than before, right? Solution, as you know, as all of you may know, deep learning has changing everything, right? So one point is anyway how the business model has been changed in terms of antibody drug discovery. Another purpose of my talk is to this one, to overestimation and underestimation of AI, case correction. This is a risk, because my current perspective is in this era of industrial revolution driven by deep learning, it is crucial to adapt and fully leverage the benefits of this revolution. Okay, to achieve this, it is essential to avoid both overestimating and underestimating AI across different fields and to foster mutual understanding. That's why, okay, the second purpose of my talk is to present key discussions on deep learning that may also be useful for those who are not familiar with antibody drug discovery. Okay, so anyway, two purposes. Okay, let me start. So before my main talks, I would like to introduce a little bit about our center, RCCS, which is a similar organization of VSC, but which is a research center out of 13 centers in Riken. Okay, so it's located. The headquarters of RCCS is located at Kobe, all right, the west side, west field area of Japan. And we have also the magnificent building with power generation. We are constantly visited by the VIPs all over the world. In this case, Emperor and Empress in Japan. And we're happy to let you know that, okay, so we operate the supercomputer Fugaku, right? And we're happy to let you know the industry user of Fugaku is increasing, okay? So Japan is strong at heart simulation, very accurate heart simulation, and also the flow simulation in the cardiovascular. These two are the very good companies, right? So, and another company called Adivexis has a AI-based computational drug discovery company. They are also using the Fugaku. But anyway, RCC is not just operating the Fugaku, but they are plenty of renowned researchers all over the field, and we have the global collaboration, the partners all over the world. Sometimes we hold international conferences headed by Satoshi Matsuoka. He and the other scientists have plenty of awards, including Golden Bells. And one of the renowned researchers, Yasushi Okuno, who is also the professor of Kyoto University, his hope is he wants to digitalize all of the steps of the drug discovery from the beginning to the end to be digitalized. Okay, so but his project is so famous in Japan, and we are surprised that around 6 months ago, in the Japanese parliament, Prime Minister and the other 3 ministers emphasized that his project is very important as an essential as a part of Japan's national policy. So supporting these real political leaders, we are happy to construct the discovery platform. But before my joining 3 years ago, he hadn't set up he's now focusing on the discovery of small molecule. Okay, so now my joining 3 years ago, now we are setting up the discovery platform for antibody, right? So I'm a kind of playing manager. As a manager, I would promote the industry academic collaboration for all of the fields. But as a player, I'm the antibody person, antibody drug discovery person. So the contributor as a scientist, right? So again, and a little bit about Riken, I would like to introduce. Riken is Japan's largest and most comprehensive research organization for basic and applied science, and world leader in a diverse area of scientific disciplines. President is Makoto Gonokami, who was president at the University of Tokyo. And he joined 2 years ago in Riken and created a new project, TRIP, which his hope is that all 13 research centers in Riken should collaborate with each other to obtain the synergistic effect. Under the TRIP under the concept, Professor Makoto Taiji creates a new project, TRIP Aegis, using big data in Riken and using computer research in Riken to generate the foundational model of AI for science. And also, the Riken has its own drug discovery program headed by Okazaki, and also one of the associate head of the Koizumi-san. These two are previous worked at pharmaceutical companies, so they are knowledgeable about real drug discovery in industry. So I mean that also the drug discovery, like this, and AI are not only important for RSS, but important for Riken as a whole. Okay, so let me start the main topic. So so I summarize a lot of antibody research. I worked a long time. I'm just focusing on the antibody research because it's a very charming point. All right. Firstly, in addition to being a key modality in drug discovery, antibodies have a range of industry applications beyond pharmaceuticals. Secondly, they are crucial for advancing our understanding of the adaptive immune system and for eliciting efficacy and side effect of the vaccine development. Thirdly, antibodies are among the protein families with the greatest amount of sequence structure and functional information available, making them highly accessible research subjects and useful reference for studies on other protein families. So the antibody gives you the test cases for the engineers of the other protein families. Fourthly, they are the only molecules in our human body capable of rapid evolution in our body, making them uniquely suited for gaining insights into protein evolution. Fifthly, a wealth of high-throughput production and screening techniques has been evolved, making antibodies an ideal research subject for combining laboratory and in situ technology. So anyway, antibody is interesting, at least for me, right? So I love antibodies. So so I researching from 1990s, right? So the first I focus on the CDRS3 because this is the most important part. So I did the two approaches, informatics and simulation. I would like to let you introduce very quickly in terms of the antibody structure. Antibody is a complex made up of two heavy chains like this, eight chains, and two light chains, right? And it is variable regions, okay, which can bind antigen, and a constant region which doesn't bind to antigen, right? And the sync CDR loops, complementary determining regions called CDRs, bind to antigen, and among them, CDRH3 is the most important due to its diversity in length, sequence, and structures. Okay, and this is a crystal structure viewing from antigen. Okay, viewing from the antigen. CDRH3 is located at the center of the antibody, and it's the most preeminent role to recognition of antigen. Right. That's why I researching on from the informatics empirical approach and the deductive approach for simulation. Okay. After that, I joined the antibody modeling assessment. This is a kind of a CASP type competition focusing on antibody drug discovery. I would like to give it. So this competition is just J.C. Almagro, who belonged to Pfizer, one of my friends, asked 11 quizzes as a blind test. They know the antibody structures. They know the structure of 11 antibody structure, but they gave us just sequences, right? And participants are like this, CCG Axel Schrödinger, macromolecules, and John Hopkins group. John Hopkins group has a Rosette group, and we are the Japan group, right? Team Japan, right? Asteras and Osaka University, and giving us the sequence data to do the prediction of 11 structures, and they compare the sequence, compare the real structures, experiment determined, and predictions. And there are two quizzes, two types of quizzes. First, anyway, to give us all of the sequence, okay? And simply we predict all of the structure. After that, they gave us the structure, except for CDRS3. Again, I would like to say that CDRS3 is a very difficult target to predict the structure. So So the second stage is a structure, except CDR3, that's a given to us, and predict the CDR3. This is the results. Red is a stage 1, the blue is stage 2. The y-axis shows RFD, so the shorter is better. I'm proud that, to be honest, it's very lucky, to be very honest, but it's very good for me to obtain budget, all right? So I'm always advantage. So anyway, very luckily, we could win both cases, all right? Stage 1 and stage 2. So if it's training of people, also advertising this, they are the number 1 in terms of stage, but we are, we both cases are number 1, right? But so why we could achieve is just this is a paper of this paper of this competition, the high resolution modeling of antibody stress by combination of the bioinformatics. Okay, bioinformatics. Because I did very experience, right? It's very, very useful for this competition. Okay, bioinformatics, expert knowledge like me, right? Okay, too cute for this. Okay, and molecular simulations. Okay, so the bioinformatics, molecular simulation, inductive approach and deductive approach and experiment are complementary to each other, right? So the other group tend to use only informatics approach. Okay, it's a big. It would be the very the difference, all right? So the so based on the experiences, I took a read of international collaboration between academia and industry. So that single company cannot do the dramatical, how can I say, so the single company should do the competitive research, very applied research. But in terms of the basic research in the industry should be done by the pre-competitive research, which is gathering the industry and academia. So the EBI, Industrial Biometric Institute, based on Cambridge, the UK, provides us a good chance, good opportunity to do the pre-competitive research. So I raised hand and I could take a read of the antibody informatics for drug discovery, thanks to Sanofi Aventis, AstraZeneca, Meiji Immune, and IMGT group. At that time, University Montpellier group was conducting the largest antibody database. Now currently, University of Oxford group are conducting another bigger antibody database, but at that time this was the number 1. And the IEDB is a group conducting the largest antigen database group at the west coast of America, and Technical University of Denmark, right? So thanks to these collaboration, we could map the business model. This was very good because interests differ depending on the positions. President, manager, project leader, project industry, usually experiment scientists in drug discovery, of course. And in silico researcher in the industry, like me, right? Like me. And academia scientists or software developers here, right? Flow, obstacle solution in silico solution and science. This is very important. Also, please have a look in detail. So first step is the immunize antigen to the mouse to obtain hit antibody. And after that, major activity to obtain a lead antibody to be engineered to obtain optimized antibody. After PKPD detection, we obtain the antibody to be developed. After clinical trial and mass production, we finally achieve the antibody drug discovery. President and manager wants to know this step, comparing with the other project, which project they are focusing on. Okay, so which project do they continue, right? So but project leader, they are to continue to this project. So as you know, the antibody drug discovery, not only antibody discovery, as the drug discovery has plenty of hurdles, okay? So difficulty antigen prediction, antigen preparation, physicochemical properties, difficulty interpreting experiment data, okay? It's very difficult to obtain the experiment data. It's hard to understand, right? The concern immunogenicity, difficulty in prioritization. You have obtained lots of good antibodies. It's very hard to how to prioritize, right? Low activity, etc. After that, some of the obstacles can be solved by the in silico approach of the antigen design, antibody design, or rational prioritization. Hints, just hints is also important. Not always do need a design, rational design, but just hints is very, very important for understanding antibodies, all right? After that, the science is here, okay? So, this map is a very important to communicate, right? But the problem is, this is 10 years ago. Just the needs has changed, right? At that time, there was a simple antibody structure. Now the engineered type, bispecific antibody, drug conjugate, and conditional activator, all right? So the protein formulation, the protein structure is much more complex than before. So the protein engineering is much more difficult than before. And should create developability and immunogenicity. Okay, as you know, solution has dramatically revolutionary change, revolutionary everything, learning, changing the everything. All right. So this paper shocked me. All right. Why is I don't tell in the detail of this paper, but as an introduction, the authors describe here like this: protein research is still dominated by classical sequence simulation methods such as that brass and hidden models. In contrast to the domains such as computer vision, which have become dominated by deep learning. Okay, so yes, I've been the bioinformatics racer. Yes, I love the blast and the hidden models, but they recognize it's a classical, right? This shock. That's why I shocked. But anyway, not to underestimate AI. Anyway, to acquire particular technology like me, the conventional bioinformatics is a particular technology. One needs to invest years of effort, like me, yes, to develop specialized skills, and there are certain essential points that cannot be overlooked. Furthermore, even after mastering the technology, whether one continues to adhere strictly to these points is often the key to success, right? However, these critical points can also become burdens when it comes to both learning and utilizing technology, right? So the deep learning is so great. Removing these focal point, key focal points or burdens, making them unnecessary. This is why deep learning is saying they are doing the revolution, right? So anyway. This is a map of the AI technology. So it can be divided into two types: domain-specific AI and general AI. Okay, domain-specific the sequence analysis, structure analysis, and they are heavily reliant on the databases. Right? The general AI, the machine learning and deep learning. Right? Deep learning is a subset of machine learning, but nowadays deep learning is so special. Okay, so I would like to I would like to say the different classification, the deep learning is a machine learning. So the red arrows are the revolutions of these AIs. Okay, so let me start from the deep learning. How deep learning revolutions are machine learning. Okay. All right, so this contrasts conventional machine learning and deep learning, okay? And this task is from the image data to predict the name of the animal, okay? The classical machine learning is two steps. One is from the domain-specific AI of this field, computer vision field. They have knowledge, skill, know-how, experience to do the future extraction. This is a type consuming the difficult step, but once you obtain the features, you can easily use the machine learning supported by machine deep learning and the shallow neural network to predict cat, right? So the deep learning is achieve high predictability. Depends on the task, but usually deep learning is achieves the higher predictability, and they not necessary to use the feature extraction by the conventional technology. They are doing the end-to-end prediction in one process. Okay, so for decades, okay, the fields of computer vision and natural language processing fields, these scientists consider the feature extraction as a critical and specialized step. Okay, researchers have worked diligently to improve this stage. Okay, that's a fact that it's no longer needed. It's revolutionary. Okay, so so we must follow the computer vision and NLP people. So okay, like this, all right? So we must follow the computer vision and NLP people. Then how do you feel about it? The antibody? Okay, I'm the conventional antibody informatics people. Okay, okay. So you have the antibody sequence and you have a structure. You have a structure data, and you want to predict the developability. All right, developability. Roughly speaking, very roughly speaking, the stability of antibody. All right, and so that yes, conventional or classical antibody can, like me, always did very much effort to feature extraction from the to extract the sequence motif and structure features. Structure features, because the developability, for instance, we do not like antibody aggregation. Okay, it causes a problem. All right, so the hydrophobic patch is a problem. All right, so the we detect the hydrophobic patch. What's the size, shape, and number? And so anyway, yes. So we needed the knowledge, skill, know-how, experiences, all right, to extract the sequence motif and the structure features. Once we obtain these, machine learning can easily use to predict developability. Then let's consider in our field antibody feature extraction was also a vital step that requires specialists like me, right? Researchers have worked diligently to improve this stage. It seems we directly predict for antibody sequence structure information without the need for intricate human interpretation. We are going to do, right? To some extent. To check to know the ability of deep learning, what we did is whether deep learning could replace the careful inspection performed by experts. As I said, as I emphasized, expert eye is necessary. It's important. This is a human antibody structure looking from the antigen. This is a chicken antibody. Okay, so that I am a very expert of this field. Okay, so 30 years, I am always looking at antibody structure. So I can distinguish this is a human, this is a chicken. But almost all of the antibody researcher cannot detect, all right? Then my task is, can deep learning distinguish between two-dimensional images? Not three-dimensional images, two-dimensional images, just as a passport photo like this of human and animal antibodies. Okay, so we use this VAE technique commonly used to detect defects in manufacturing in the field of the computer vision field. Okay, only images of normal products. So we use only image of normal products. Normal products. This is a screw. Okay, so we. So that we must first we collect the image of the normal product of screws manufactured in factory. A large amount of them are used as input. Only normal image, and defective products are not inputted. All right. Then after the training, these AIs are trained to become experts solely on normal projects like this. Okay, so these AIs are knowledgeable of only normal project. They don't know at all about defective screw images. Okay, so after that, when images of defective projects are introduced afterwards, the AI identify them as anomalies because they are only knowledgeable about normal products. So after that defective screw image inputted, they are to us what to do. Okay, so it's a anomaly, right? So similarly, we similarly created VA trained solely with images of antibodies that are either fully fully human or humanized antibody, and which have progressed to phase 2 or beyond. I mean, I collected only human or humanized, very humanoid antibodies, and phase 2 or better. I mean the developable, to some extent developable, and humanized, right? They are knowledgeable about them. So the trend is expect to recognize only those antibodies that are developable humanoid as a normal. And this case, okay, this is a result, okay? This map, each row represents a data set. The top 3 are the humanoid antibodies. The top one is the drug or in clinical humanized antibodies. Second one is the fully human antibodies of drug or in clinical, okay? Developable and humanized. This is a human antibodies of healthy volunteer, completely human antibodies. And these 5 and the animal antibodies. And you move to the right-hand side, more anomalous, and the left moving to the left-hand side, more normal. All right. So the figure tells you that anyway, humanoid antibodies are recognized as normal, and animals are the results are varies depending on the species of animal, but tend to recognize the anomal. So that in the case of a chicken, this is an anomal, right? Please have a look at again. This is the human antibodies, developable antibodies. This is human healthy volunteer antibodies and chicken antibodies, and this is the rat antibody. Rat antibodies are relatively recognized as normal. Okay, so you very much we are very much familiar with antibody sequence. Yes, rat antibody is somewhat similar to human antibody. So it's natural. Okay, so again, I would like to emphasize, I'm very expert of this field, so I can distinguish, but normally it's very difficult. That's why, so AI, so it's so great, okay? Okay, so AI skipped my 30 years experience. Oh my God, for me, oh my Buddha for me. Okay, anyway, so I mean the end-to-end prediction or eliminates the burden of machine learning, also the virtual eye by deep learning eliminates knowledge of structural biology, right? In terms of structure analysis, right? Then I would like to move on to the other part. So these two are my first generative AI, generative AI for antibody sequences. This is used the CNN GAM method, which revolutionized the computer vision field, and excuse me, and this is also. Two different types of generative model I used. I generated, all right, and in this case, starting from the very random sequences, very random, okay. So like the computer vision field, they are also starting from a very random sequence, very random feature to obtain. Finally, they obtain the image very similar to the true image. So they can generate the fake image, which is very similar to true from the random images. We also, from the random sequences, so finally we obtained starting from QVQL and TVSS like the very similar antibody sequence which is obtained by the experimentally, right? Another method which is RNN LSTM is a method which revolutionizes the natural language processing field. In this case, not starting from the random image, but starting from the antibody-like sequences, but the length is a variable, like the length of the input sequence is a variable of their NLP method. And finally, finally, it's the similar way obtains a very reliable, very realistic antibody sequences. Okay, so anyway, these methods are very useful for the design of phage display technology, and this the similar thing can be done by the conventional bioinformatics. We can do that without deep learning, but to do that, very difficult to say that anyway, to obtain bioinformatics skill and to use always meticulous or the burdensome, intricate technology and is necessary. But it's very easy to construct and to use, right? So as you know, the multiple alignment was necessary when I used the conventional bioinformatics. But you collect plenty of sequences. It's difficult to align multiple alignment. But deep learning is not necessary to do the multiple alignments. It's one of the big merit. Okay, so, and another thing is the data augment. Okay, I would like to skip, but anyway, deep learning is the revolutionized and by removing the focal point, okay? And as you may know, the deep learning, it changes itself revolutionary, right? And like this, from the classical deep learning needs a level of big data necessary. But now, as you know, the training fine-tuning model, so not more already you can construct a deep learning model with a small level data based on the shared pre-trained model, which can be constructed by unleveled data, right? So anyway, and okay, I would like to skip. So anyway, I would like to emphasize that don't underestimate AI, because revolutionary everything of the domestic AI and general AI. But the limitation is like this, right? AI is a merely inductive approach, okay? Although I said I emphasize AI is great, deep learning is so great, we might feel as though AI is extrapolating, but this is merely an illusion, okay, caused by the dense clustering of big data, okay? What they are actually doing is interpolation. AI is weak in handling unexpected or highly specific cases, right? Since antibodies possess high selectivity, each individual antibody has its unique characteristics. So antibody case is a highly specific case. Okay, so it causes intrinsic or difficulty tackling with by the AI, right? And another important point is protein science, including antibody engineering, is fundamentally different from the fields of computer vision and natural language processing, in that it is governed by physical chemistry. Okay, it's dramatically different, dramatic difference between these areas and protein science. Okay, so still I need deductive approach, right? Also, inductive approach, we must follow CV and NLP field because it's very convenient, but the deductive approach is complementary, right? So the MMMD and free energy calculation, okay? So, okay, what I did achieve recently is like this, okay? So let me show the types of aggregation. This is the protein aggregation can lead to side effects of and reduced activity. So aggregation is a very bad thing, right? And there are two types of aggregation. One is reversible aggregation, like this, okay? Where proteins come together in their native structure, okay? It's not so awful, okay? But this type is terrible. The other is irreversible aggregation, which happens through partial unfolding. The latter is more problematic. So it is important to prevent proteins from unfolding. Okay, unfolding is very important if you even if you consider aggregation. So what our task is how to prevent partial unfolding. Okay, so what we did is can we learn from the results of experiments conducted over geometrical, geological time. Sorry. Okay. So let's consider, okay, proteins derived from some fillers. Okay, proteins derived from some fillers are maybe considered as extremely stable proteins that have withstood accelerated testing over geological time periods. All right, they are very very so they are the results of the accelerated testing over many years, right? And the proteins from the other species are not the usual. Okay, so the in silico way we thought that by finding MD simulation conditions that would more normally break down. Okay, like this. But not those, not break down by the proteins thermophilic bacteria. We might uncover the mechanism behind the stability, right? So to do this, this is a preliminary result outer-short simulation norm I named. Okay, so we picked up all structures, all representative structures. You go to the SCOP database. The SCOP database classifies all of the protein structure types, and we picked up the one structure representative from all of the structure, and more than 10,000. And some fillers domains is around 1,000, and the other domains around 10,000. And we did we gradually increased the temperature from 300 Kelvin to around 400 Kelvin. All right, very short second, because many structures, so it's very difficult to compare to use supercomputer. But anyway, the y-axis shows the Q-value degree of unfold. Okay, if you compare these ones, the base one, you can see a clear difference for me, at least for me, all right, to some extent, all right. And so to do this, AI cannot achieve this one, all right. So the MD simulation is very usually used this kind of tackling, all right. The last thing I would like to show is, okay, so the reality, I would like to show the reality at the discovery site is that, okay? So if we are now in industry, okay, industry, which is for experimental researchers like this, so I obtain an antibody, okay? I want to antibody to be developed, but the problem is a bit unstable, a bit stable. They want to improve TM. Otherwise, they need to restart from the beginning, okay? So their wish is to improve TM as quickly as possible, okay? Otherwise, the manager scolded us to terminate the project, right? As significantly as possible. With minimizing the reduction of affinity, of course, with minimizing the risk of increasing immunogenicity, with a small number we experiment, all right? These are the real wishes in industry, all right? Okay? So the ultimate challenge in Asteros, I belong to Asteros, is point mutation would be the minimize the risk of increasing the immunogenicity and significantly and quickly improve TM while minimize the reduction of affinity in a small number of experiment. Okay, this is the ultimate challenge. We did this one and achieved that one. And we could publish a paper last year. The first name is Yusuke Tomimoto. And also the excellent work with Riken Yamazaki. But this stability score, this is presented by Chemical Computing Group, software called MOWE. And they, the delta G calculation, delta G is a change of some strategy with a mutation, okay? Delta G unfold and fold, the difference of fold and unfold, some stability of a protein. So delta G is a mutant native change, all right, of some stability of a mutation, okay? So that we want to just have single mutation and significantly quickly improve TM while minimizing the deduction of affinity in a small number of experiments. Can do that? We could do that. But the first thing we did is a comprehensive RNA scan. So that Kumamoto and Riken-san did that all of the amino acids, all of the positions were changed to learning and compare the prediction x-axis and real melting temperature in experimental determined melting temperature y-axis. As you know, is there a correlation between TM and this stability scale? No, very low, and nothing. I can say nothing. All right. So don't be pessimistic. So that simple comparison of the RNA scale, comprehensive mutation, there's a no or at least a very low correlation. But if we do like this, okay, two-step methodology could achieve melting temperature. We calculate the stability for mutation to all 19 amino acids at every position. So it's very easy because the computer we can to do that by experiment is terrible. In this case, VHH antibody has around 100 residues. Okay, so 100 residues maximize 19 mutations. 1,900 experimental mutation is a very table, but in silico it's easy to calculate, right? And after that, select step mutant, which shows that the in silico ranking, right? And measure TM on only 5 mutants. We did the experimental research to measure TM for the type of mutants by experiment, and we actually 3 out of 5 increased TM. Okay, interesting. Okay, so there is a very little correlation, but few correlation. But the top mutant actually 3 out of 5 increased TM. Okay, and 1 increased more than 5 degrees Celsius. It's splendid. Okay, after the position increased 5 degrees here, right? So after that, we change remaining 80 amino acid and measure TM by experiment. 5 increased TM more than 5 degrees. Okay, so anyway, if you this one, if you this technology, so the mere comparison, I can say, general comparison is very terrible, but you select only the very top level prediction, you can actually experiment, enhance the TM. It's interesting, right? So anyway, deductive approach, I show you that this case, and this case can be achieved not by AI, but deductive approach. Okay, so I would like, so let me summarize by 2 slides. Today I want to tell you about two points. One is how the business model changed, okay? So needs are very changed, dramatically difficult than before, and the solutions has very changed in terms of especially inductive approach. Deep learning is changing everything, but just limited on the inductive approach. We have to compensate with the deductive approach. Okay, this is the point. And the conclusion of this is a general purpose, not underestimate. Deep learning has eliminated the need for the feature steps. So what was in that's essentially in traditional machine learning, general AI allows us to avoid the specialized and cumbersome tasks that were once necessary in bioinformatics. The use of virtual lines means that in-depth structure analysis is no longer essential. Okay, so not to overestimated simulation approaches complement AI's weak in handling unexpected high specific cases. Since antibodies possess high selectivity, each individual antibody has its unique characteristics. Antibody molecules are governed by the laws of physical chemistry, and each antibody has a unique characteristic. Let me visit the acknowledgement of Riken people and asterisk spam. So thank you for the patience. Please forgive me. Always I have a problem with English as a foreign language. Sorry, but anyway, let me visit. Thank you very much.
Don't worry, I think we understood you perfectly well. So there's time for a few questions. If not, I have multiple, but please, I'll come down.
Thank you for your presentation. You mentioned CASP competition.
Yes.
And maybe it's kind of annoying questions that people ask you all the time. AlphaFold.
Yes.
AlphaFold, what do you think about evolution of methods of AlphaFold 1, 2, and 3? Because in AlphaFold 1, they were using convolutional neural networks that you mentioned. In AlphaFold 2 in 2020, that's one with like even bigger margin. It was convolutional neural network and transformers. And AlphaFold 3, we don't know. Transformers for sure, but probably based on description of the paper, it's graph neural networks.
Yes.
What do you think will be next? What do you expect?
Yes. All right. So yes, as many research, many science surprised, I also very much surprised in terms of AlphaFold 2. Okay, AlphaFold 2 dramatic changes AlphaFold 1, right? Okay, and furthermore, AlphaFold 3 diffusion model dramatic changes AlphaFold 2. Okay, so and if you have looked at the paper of AlphaFold 3, just we are testing it with Riken, of course. Now, yes, it's very splendid, and but still they are as they recognize, still antibody loop modeling is challenging. Okay, it's much more accurate than before, but still problem in terms of the AlphaFold 3. So I think convolution network. So transformer. Every deep learning model is changing to the transformer model, and probably BSS or RCC also is tackling with the next transformer model. Okay, probably the paper will tell you about the attention is not at all your need or something like that, right? So, but at this moment, if you look today, today I didn't summarize, but if you do the antibody modeling by AlphaFold 3, still has a problem of the loop regions. Because, as you know, the deep, so actually CDRS3 is, in the case with CDRS3 is that intrinsically disordered regions, okay? So the goal is not a single structure, right? So the latter part of your question is what we need is anyway to identify the plural structures depend on the situation in our body and in the condensed structure, in the diluted structure. It's still far away. Okay, anyway, we need to use the convolution network RNN as the over in my feeling. All right, transformer or the next model of the transformer in combined with physical approach is necessary to do that. Yes, in my opinion. Okay. Yes.
So, have you tried physical informed neural networks? Because you can modify loss function by some like if we are talking about physics.
Oh,
So you incorporate some like equations to your like constraints to training of your neural network.
yeah. No. For me, I'm not. Yes, many people are trying, but I find myself not yet. Right. Mm.
Let's change group and then we'll go back. Obviously, you have lunch with him, so together.
Thank you for the presentation. My question is more in the immunology focus. Could you give us, if you know, an insight of the participation of deep learning and machine learning in CAR T cell therapies?
CAR T cell, yes. As you know, yes, now the immuno-oncology is so interesting, isn't it? Right? The dramatic changes the cancer therapy, right? So at this moment, the trend is one thing is to find the new epitope. Okay, so as you know, the MHC, so the prediction of MHC and peptide is almost over. Now the complex, so how to recognize the T cell receptor bind to peptide MHC complex. Okay, so this is a challenge of the AI. Okay, so the people in the immunology field are trying to collect the big data. But one problem is T cell receptor is not so diverse because antibody has a diverse because the somatic hypothesis mutation occurs. T cell doesn't occur, right? So the interaction is so weak. Okay, weak. That's a problem. But anyway, the key point is how to find a new epitope. And another point is how to recognize efficiently the T cell. In such a case, you can do the so yes, so CAR T therapy, right? CAR T therapy, right? So change the good, so the better artificial T cell epitope incorporated with that T cell, right? Okay.
So thanks for the talk. I like the slide that you showed how the information flows from the academia side to the industry side.
Yes.
So my question here would be, what are the questions that you think that computational scientists as us could help on?
Yes. Anyway, so now, yeah, this is the view of 10 years ago. At this time, it's very separate. So academic science tend to do the basic research, all right? And industry research, of course, just approach. But not like Professor, he's great, both knowledgeable industry and science, basic. But anyway, so if you belong, so you belong to the academic side. Now you need to at this moment AlphaFold 3 almost finalizing the modeling. So the next step is I think one thing is generative model for generating for the de novo design. This is a challenge, but a very important point. And another thing is, anyway, to remove the immunogenicity and enhance developability, and also the affinity, to enhance affinity with a small number of experiments, right? This is a downstream task, all right?
But the thing is that for doing this task, mainly you need like specific databases that you have antibody sequence and then the antigen sequence or the epitope region. So
yes.
I think that to be able to reach that point for the case of antibody binding, we still need more databases that are more extended, because usually what you find is like databases of antibody antigen without the antigen.
Yes. So depends on the task. I think yes. So that if you classify what the structure-based approach, like probably your method is a structure-based method. If I'm correct, and LLM method can be can be differentiated. Although they tend to be joined, but if you classify, so the LL method is very hard to do the de novo design because shortage of the data, big data, right? So if you do the developability, you can. I think you have plenty of data. It depends on the task, right? Yes. Excuse me.
ありがとう。
ありがとうございます。Yes.
And the last question for today.
Thank you for your talk. I was going to ask you something about the modeling of CDRs. I'm following to what he said before.
Yes.
Generally, when we measure the accuracy of generative AI to model the CDR structures, we use structure metrics. But in a few projects, we've realized that another good option would be to test for the maintenance of the interactions between the CDRs with the antigens. So I wanted to hear your point on that.
Both are important. I think it's now the challenge. Yes, the combination is always changing, right? So both are necessary. It's I'm sorry, I don't have a concrete answer. I want to challenge both types, right? Yeah.
Okay.
Okay. Thank you.
Sorry, and thank you. Do you have any other safety endpoint that you would model for antibodies or only immunogenicity?
So sorry?
Do you have any other safety-related endpoint that you would model or predict for antibodies or only immunogenicity at the moment?
Immunogenicity by experiment. So this January, a genetic group had reported a very splendid work reviewer. But as you know, immunogenicity causes by plenty of very complex. So humanization is one thing, right? Humanization is one, and to eliminate the T cell epitope is another thing, and so the immunogenicity, the B cell epitope, so eliminates the B cell epitope. Immunogenicity is one thing, right? One thing. Another point is that the host cell, the contamination of host cell protein, very different. The analytical people, right? And sometimes depends on the medication and the disease. So anyway. The thing we need you to do the various types of phenomena, okay, so that not a single approach can achieve this. So I don't like some scientific paper or some venture companies say that their single approach can achieve all of them. I don't think that. So it's a very complex field. Yes. Right.
Thank you.
You're welcome.
All right, so well, you see, it's a hot topic and people are quite interested. And I think your point is well taken, that it's in AI. We know it's interpolation plus a little bit of randomization. You can make it probably more random, but of course, when you interpolate in something where a single mutation disrupts completely the dynamics or the snapshots of the CD3, then everything goes away. And we see a lot in it's very different to cherry pick. Like in most retrospective benchmark, cherry pick the good ones when there are good ones, but when you're trying to do this prospectively, it's a completely new world, and that's where the difficulty is. So yes. We'll see. So thank you very much, Hiroki-san. Extended talk.
Thank you very much.